In Part 1, we mapped the electron transport chain, from NADH to oxygen, from Krebs cycle to ATP synthase, and we showed where methylene blue sits in the thermodynamic landscape of that system. Now the question is, what happens when electron flow at that level is improved? What are the downstream biological consequences? And where does the evidence for those consequences actually come from? Virtually every chronic disease shares at least 2 of 3 common biochemical features: an ATP deficit at the cellular level, upregulated systemic inflammation, and impaired insulin signaling. These are not separate problems that happen to co-occur. They are mechanistically connected, and the connection runs through mitochondrial function. When electrons leak at complex 1 and complex 3, they react with oxygen to form superoxide, the initial reactive oxygen species. Superoxide feeds into downstream ROS cascades. Excessive ROS activates NF-kappa B, the master transcription factor for inflammatory gene expression. NF-kappa B upregulates interleukin-6, interleukin-1 beta, and tumor necrosis factor alpha. Inflammation escalates. Simultaneously, impaired mitochondrial function forces cells to rely more heavily on anaerobic glycolysis, the less efficient cytoplasmic energy pathway. This shifts the cellular metabolic state in a direction associated with insulin resistance. AMPK, the cellular energy sensor activated when ATP falls and AMP rises, is underactivated, and insulin signaling downstream of AMPK is impaired. 3 failure modes. 1 root, electron leakage at the mitochondrial inner membrane. The brain accounts for 2% of body weight and consumes 20% of all oxygen and 25% of all glucose. Neurons are the most metabolically demanding cells in the body, with the possible exception of photoreceptors, which we will get to. ATP production in neurons powers 3 things specifically: maintaining the sodium-potassium pump, synthesizing neurotransmitters, and sustaining synaptic signaling. Any of these functions fails when ATP falls below threshold. Neurological conditions including Parkinson's, Alzheimer's, and depression share one documented feature: mitochondrial dysfunction in neurons. The precise mechanisms differ by condition, but the common denominator is impaired ATP production in cells that cannot tolerate impaired ATP production. A 2014 study in the Journal of Alzheimer's Disease documented that methylene blue reduces tau protein aggregation, the neurofibrillary tangles characteristic of Alzheimer's pathology, by 50%. The proposed mechanism is not direct chemical interference, but improved mitochondrial function. More ATP available means healthier protein quality control and less oxidative stress. A 2010 study in neurobiology of aging showed methylene blue increased brain glucose utilization by 30% in age animals and restored memory retention. A 2016 study in the Journal of Psychopharmacology documented a 10% improvement in memory consolidation from a single 1 milligram dose in healthy humans. Not a week of treatment. 1 dose. The retina is a thin sheet of neurons. Its photoreceptors, the rods and cones, have the highest metabolic rate of any tissue in the human body, higher than cardiac muscle, higher than neurons in the cortex. They run at near maximum capacity continuously, powered by dense mitochondria. 2 of the most common causes of vision loss, glaucoma and diabetic retinopathy, involve mitochondrial dysfunction and oxidative damage to retinal ganglion cells. The 2017 British Journal of Ophthalmology study showed that methylene blue, by improving mitochondrial function and reducing ROS, protects retinal ganglion cells from death. The 2015 PLOS1 study showed a 60% reduction in retinal damage in diabetic models. Same mechanism, optimized ATP production, reduced oxidative stress. The clinical weight of this is significant because retinal cells cannot regenerate. The human retina has no meaningful regenerative capacity. Every ganglion cell lost is permanently lost. The mechanism by which any intervention might protect these cells before they die is therefore of genuine scientific and clinical interest. In 1931, Otto Warburg won the Nobel Prize in Physiology or Medicine for documenting what is now called the Warburg effect. Cancer cells preferentially use glycolysis to generate energy even in the presence of oxygen. Normal cells use oxidative phosphorylation when oxygen is available because it is far more efficient. A 2019 paper in Frontiers in Oncology proposed a mechanism. By normalizing mitochondrial function in cancer cells, it forces them toward oxidative phosphorylation. Cancer cells with dysfunctional mitochondria cannot sustain this shift and undergo apoptosis. Healthy cells with functional mitochondria handle the transition easily. Whether it translates to clinical oncology outcomes is a separate question that requires clinical trial data. The mechanism is there. The clinical validation is not yet complete. Methylene blue is a pharmacologically active compound. It is not a vitamin. The following is a summary of the most significant known interactions and contraindications. The most serious known interaction involves serotonergic medications. At doses above 1 mg per kilogram, methylene blue functions as a monoamine oxidase inhibitor. Combined with SSRIs, SNRIs, or tramadol, this creates the conditions for serotonin syndrome, a potentially fatal state of serotonin toxicity. This is not a theoretical risk. G6PD deficiency, a common genetic variant, impairs a cell's ability to manage oxidative stress. Methylene blue triggers hemolytic anemia in approximately 70% of G6PD deficient individuals. Pregnancy and breastfeeding are absolute contraindications given the absence of safety data. Chronic high-dose use is also mechanistically counterproductive. At elevated concentrations, methylene blue shifts from antioxidant to pro-oxidant behavior, increasing oxidative stress. The evidence supports a cycling protocol, not continuous daily use. A molecule synthesized to color cloth in 1876. Its reduction potential, positive 0.01 volts. It fits precisely where thermodynamics favors electron flow. The electron transport chain has been operating for 2 billion years. Methylene blue fits it the way a key fits a lock that was built before the key was made. Whether that property translates into outcomes is a question that requires a physician, not a video. What it requires from us is precision, not hype. The mechanism is documented. The biology is real. What you do with that information is your decision to make, ideally with someone who knows your full medical picture.